Well, good morning, everyone. So I really appreciate you're here. So my name is Santiago Schnell. I'm the new Dean of the College of Science. So the College of Science Dean is called the Warren K. Williams Foundation Dean. And I would like to welcome to the University of Notre Dame and this great football weekend. You know, one of the things that really impressed me from the University of Notre Dame, I have held faculty positions in different institutions, is that when people come and ask the question, what makes a football weekend in Notre Dame so special? Everyone imagines that it's exclusively the excitement of the game, the rivalry between the teams, that actually they get the best of each other at the field. But the reality is much, much bigger. So, and it's much more exciting. So we hold a series of events to celebrate with our families and friends what the University of Notre Dame has to offer. And it's not only what it has to offer in the campus, which is gorgeous, but as well in the classroom and in our research laboratories. And you know, for me, I want to welcome you for this special occasion because we're inaugurating the Science Exploration Series today. We have a wonderful series of speakers this semester. They're representing different departments from the college. They are going to be talking about their groundbreaking research from the farthest corners of the universe. We have people who work on the origin of the universe. And then we have others that work on very complicated issues inside the cells. We'll have a series then on Friday, this next Friday, in Chicago, that is going to be held before the Shamrock series. So if you're going to be in Chicago next Friday, so I would like to encourage you to join us on Friday. We will have the next lecture there. And today, I'm very pleased to introduce the director of our DNA Learning Center, Dr. Abby Stark. Our DNA Learning Center opened to the public in 2015. It has been growing ever since. Just to give you an idea of the growth, the center offers a hands-on experience where children, they can get out of the classroom from their schools, come into the laboratory and have a hands-on experience on how the state-of-the-art molecular biology is done in our laboratories. So we have reached over 200 groups, and that's equivalent of close to 9,000 people since 2015. Unfortunately, the pandemic played a role on stopping the program, and those hands offerings were suspended on March 2020. But the exciting thing is that we're opening this one, and I'm really thoroughly excited to have again students across the state of Indiana and the biggest Great Lakes area to come in and enjoy the experience of doing science at Notre Dame. In addition of being the director of the DNA Center, Dr. Stark has been the executive director of the Northern Indiana Regional Science and Engineering Fair that is actually held on the campus of the University of Notre Dame every year, and she has been directing this initiative since 2018. She has her PhD in human genetics from the University of Chicago, where apparently funds goes to die, that's what they say. And she has a passion for outreach and expertise in genetics and biomedical research. Very few scientists actually would like to engage with the public, and she's not only one of those few who are an exception, but at the same time, she's exceptional on her job. In addition to that, she has held a number of positions in the American Society for Human Genetics. So I want to thank you very much for coming here and supporting science at Notre Dame, but at the same time, I want just to give the welcome to Dr. Stark. Thank you very much.
Thank you. Well, good morning. I am excited to be here today. I know for some people attend the Science Exploration Series every week. So I know last week you were in the planetarium and got that type of experience to be able to see the stars. And so because the DNA Center is a super hands-on oriented center, wanted to try and have some part that's not just you listening to me talk, but actually be able to do some science as well. So for the second half of this presentation, you guys will actually be able to isolate and precipitate your own DNA and be able to visualize it if you're comfortable doing so. So while I'm up here talking, Ms. Kim from the DNA Center staff will be distributing some supplies that you'll need for the second half of the talk. It will require you to lower your mask briefly to be able to do that because you're going to have to collect your own DNA to be able to precipitate it out. And so if you're uncomfortable with that, that's totally fine. If you just want to like maybe discreetly shake your head so we'll know not to leave supplies by you, or if you change your mind, that's also fine as well. So I'm going to talk at the beginning a little bit about how DNA is used in society, which I think if you read the little synopsis for today, I'm going to hit those three main topics about 23andMe, Ancestry.com, sort of forensics that you see on TV shows, and then of course the coronavirus, which if you weren't a scientist before, you've gotten an inundation of all kinds of different science there, but we're going to focus on just the genetics of that. So I'm going to talk for a little bit at the beginning about those things, and then we'll turn it over so that you actually are able to do some science yourself, which you might be surprised on how straightforward and easy it is to precipitate your DNA. And hopefully after this, you could actually go home and do this with your kids, grandkids, neighbors, whomever you might want to be able to show their DNA to. It's kind of a neat experience, and it's pretty straightforward. You can use all the supplies you have in your house, which we'll talk about once we get there. So as Dean Chanel said, I do have my background in human genetics. It's always, I think, nice to know who's talking to you, so you know, do they really know what they're talking about, or are they just sort of reading something that you can find on Wikipedia? And so I do have my PhD in human genetics, so I approach everything kind of from a genetics mindset. And obviously I'm very biased since I decided to get my doctorate in that, but I think genetics is sort of the most exciting field in science because it's such a part of everyone's life. You can't miss it, right? Even if you never took a science class, you have DNA in your life with the commercials that you see for 23andMe and Ancestry.com. Lots of local public libraries have genealogy sections now, and they bring DNA into it. If you have anyone who has any type of health concern, a lot of times you're doing genetic testing there. When you're pregnant, there's a lot of prenatal DNA testing that you can decide to do or not. And if you're uncomfortable or you don't have sort of a baseline genetic literacy, it can be really hard to know what you want to do in those different scenarios. And so one of the goals of the center is sort of to just bring genetic literacy to any group that is interested in learning a little bit more about it. Not to become geneticists, but to be a little bit more comfortable being able to talk about those topics. And so, as Dean Chanel said, we do field trips, but we also do adult professional development events. We've had after school clubs. We've had a hockey team from Wasilla come. I think they were here on campus for a tournament anyway, but otherwise it'd be a really long trip to come just for the DNA Center. But it is a really unique and fun experience. So I would encourage you to spread the word out to your contacts once you head out. So we're going to start by talking about 23andMe and Ancestry.com commercials. Probably everyone in here has seen them a lot. They're sort of all over the place. You can sell the kits in Target now, and I think possibly Walgreens and CVS as well. And so just for kind of fun, how many of you have done 23andMe or Ancestry.com already? All right, so maybe about 20% or so. And the rest of you obviously haven't. So one of the questions that I always get when someone finds out that I'm a geneticist, there's a couple of them when they've done 23andMe and Ancestry.com, but one of them is always, is it real science? Like, is it true? And so that answer is super easy. Yes, it's real science. It is true. And so the nuances can sometimes be in how you interpret it. And sometimes people are confused because you'll get emails that'll say, you've got an updated report, or you've gotten your ancestry, maybe ratios have changed just a little bit. And so that can be confusing because why would it change if it was real, right? If your ancestry doesn't change, you know, your ancestors are your ancestors. And so why should your genetic report change over time? And the reason for that is because the knowledge base is continuing to evolve. Right. And so the reference that the companies are using to be able to annotate the ancestry is evolving. And if we had a database that had the exact genetic profile from every ancestor, every country covering the earth, it wouldn't change, right? It would be a comprehensive analysis. but that is not practical. And there are certain regions of the world and certain ancestries that are maybe better represented, they're better queried in their reference panel. And so it's a little bit more accurate for those than ones that don't have a whole lot of reference sequences in. And as they gain more reference sequences, that's why some of those reports change over time. It's not that your ancestry has changed. Your ancestry hasn't changed, right? Your ancestors are your ancestors, but it has evolved as those reference panels evolve. And so sometimes it can be really hard to think about how can you use DNA to map out where ancestors are coming from, especially when we're talking about generations ago. And so if you think about it, we have 23 pairs of chromosomes, right? You get one set from your mom, one set from your dad. Pretty much everyone remembers that from basic science classes, right? And so when you think about, we'll just pick the paternal lineage for a second. That one chromosome you get from your dad is a mixture of the two chromosomes from his mom and his dad. He doesn't get a perfect copy of one of those. It kind of gets mixed up and he gets pieces from, each chromosome has pieces from his mom and pieces from his dad, right? And if you extrapolate that back another generation, his dad is a mixture of his two parents, right? And so as you go back and back and back in the subsequent generations, those pieces of the chromosome that sort of got shifted around, even though the chromosome looks like one solid piece, it comes from different ancestors through a lot of different mechanisms that you don't really want to learn about, recombination and all of those. But basically what it means is when you start looking at a base pair by base pair, you can start to map regions of the chromosome that came from different ancestors and come from different parts of the world when comparing it to the reference sequence. And so as those reference sequences evolve, the map might change a little bit like we just talked about, but that's how you can do it by looking at a base by base, letter by letter sequence. You can start to see, oh, this piece comes from your great, 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 great grandfather on your mother's side. This piece came from your great, great grandmother on your paternal side. And that, obviously, again, I'm very biased, but I think it's super neat that you can actually do that, right? As opposed to looking at your chromosomes and saying, well, this chromosome came from my mom, this chromosome came from my dad. That's true, but those chromosomes had to come from somewhere as well, right? And so the second question people get when they do Ancestry.com is they say, so what? What can I do with this information? This is so, I have this great report, what do I do now? So that varies by what you want to do with that information, right? So everyone gets that ancestry information, and some people go off on that and do genealogy and start researching different parts of the world and finding ancestors and doing things like that. A lot of times you can also take the raw data file and go to secondary providers and get sort of different reports to find out about your health and wellness and metabolism and how you process different chemicals and all kinds of things like that. And so the reliability of those reports vary. So almost everything that people distribute to you is scientifically accurate. There might be an association with a genetic marker with a health or wellness trait. But the degree of which that single genetic marker defines that trait is often not super high. We're not talking about, some of those disease-causing genetic variants where if you have the genetic marker, you get the disease. These are things that maybe increase your risk a little bit, but you have to really kind of have a sophisticated understanding of risk, because I had one person, she was actually a photographer. She was taking pictures at an event and she heard I was a geneticist and she was super excited. She goes, I just did 23andMe and I found out I was at high risk for developing blood clots. So it was super useful because now when I'm working at my computer, I set my alarm and every hour I stand up and walk around so that I don't develop blood clots. And she was like very, very serious when she was saying this. And I was looking at her inside and I didn't want to discourage her, but I was like, that's not what that report means that you need to do, right? Because saying that you're at an increased risk, even a much increased risk of something that is still exceedingly rare, is not something that you need to change your day-to-day habits for, right? It's something to maybe be cognizant of. Maybe that would be something she'd be worried about if she was taking a flight, a 22-hour flight or something like that. But it's not something that you need to change your day-to-day basis. And that's where a lot of the criticism for DTC genetics comes from, is from people sort of misunderstanding some of the reports, making lifestyle changes, and then people thinking, well, they didn't really understand what they had just learned, and so people shouldn't get access to that information because they can't understand it. And I think that's a little bit of an overreaction too, because you just have to improve the communication and the education. But I am always a big proponent of more information is better than less information, right? And so if you can get that access to that information, I'm always supportive of that. And a great thing about 23andMe and Ancestry.com is they do give you the raw files. So you are able to query it yourself, should you choose to do that. So if you see a big article that hits, the popular press about a genetic variant that's linked to Alzheimer's or Parkinson's or cancer or something like that, you can actually go into your 23andMe and Ancestry.com file and see, like, do I have that variant? Even though it's not a one-to-one relationship generally, it still sometimes can be interesting just to know what that. So it's more of an interesting thing than it is sort of, you should make huge lifestyle changes because of it, right? And then let's see, the third thing that people generally sometimes get a little bit hung up on those tests is that genealogy and ancestry are sometimes used kind of interchangeably, but ancestry, or at least genetic ancestry, really is talking about where your DNA comes from. And that doesn't always match up with where you identify socially, right? If you grew up Italian and maybe you come out and you're only 24% Italian, that doesn't change the fact that you think you're Italian and you make the world's best gravy for your spaghetti and have a family recipe that's been in your family for generations to make the best lasagna in the world, right? Those things don't necessarily change your identity. And that's another criticism. And this is even one of the commercials, I think Ancestry, or I don't remember if it's 23andMe or Ancestry has, where someone discovers something new in their genetic report. And so they get rid of, I think, like their kilt and they go buy lederhosen or something like that. That is a little bit of an overreaction, right? Because how you identify socially can be very different from what your genetics is saying. And it doesn't mean you should, throw away your gravy recipe that's been in your family for, generations because you turn out to only be 18% Italian when you get right down to it. There's lots of reasons why sort of social genealogy and ancestry might not match up with genetic ancestry. And that's probably a topic for another talk on why those two things might be a little bit discordant. But it doesn't mean, just because you learn about your genetic ancestry doesn't mean you have to toss the genealogy side and your social sort of identity aside. And that can sometimes be a little bit of a hang up for people, but I encourage you to think about it sort of as two distinct pieces of information that are sort of complementary, but they don't need to, replace one or the other. And so this kind of, I'm going to connect right in to the forensic application of DNA because it kind of, you'll see how it links in a second, but you probably, if you watch TV at all, have seen shows that use forensics, right? There are some shows that are almost exclusively dedicated to it. Some shows, it's a component of it, right? But I think CSI sort of started that trend, and now they're making a remake of it coming out this fall. So, it's been so long that it's been remade. But that is, using forensics to solve crimes has been around for a really long time, right? Fingerprint analysis, well before we could do any type of molecular analysis and things like that. But for the most part, forensic applications of especially molecular biology and genetics tends to be sort of behind the research sphere, which makes sense, right? Research has to figure it out, figure out all of the kinks and things. And once it's really smooth, then it gets applied into the legal realm. And so what that means is on TV shows, almost always what you're seeing is real science. It's not science fiction. Sometimes they mispronounce things, which kind of makes me giggle a little bit when they're like, just have sort of like the wrong, emphasis on the wrong syllable. Like you could tell it's the person who's saying the words aren't familiar with what they're talking about. But the actual presentation of science there is generally pretty good, whether we're talking about, NCIS or some of the more fun TV shows, or something like Forensic Files, where it's a lot more, scientifically focused. But the science there is generally pretty good. And one of the things that's starting to be on a lot of the shows is this idea of doing sort of forensic ancestry. And this idea has been around, like the science has been there for a while. You can actually take a genetic profile and extrapolate sort of the physical traits of that person without seeing them, just looking at their DNA. And so forensic scientists thought, well, one of the limitations of using DNA in forensics is you have to have a suspect to compare the profile to, right? It doesn't do you a lot of good if you've got a DNA sample and you don't have anyone to compare it with. And so the question was, well, how can we use that DNA sample to help us identify someone who committed a horrendous crime? So you can use it primarily two ways. One, as I just described, with the traits, trying to find out what is their likely skin tone, their hair color, their eye color. You can start to build sort of a physical profile of your suspect. And this has been in the news quite a bit. I think there's a serial killer in California that got connected with. You can also do forensic ancestry, where you can take your suspect's DNA and you put it into a lot of the publicly available ancestry databases, not with the assumption that your suspect will be in there, although if it was, that would be even better, but with the idea that you will identify their third, fourth, fifth cousins on each side of the family. And then you can hire actual sort of statistical geneticists that have usually been used to try to map sort of like the origins of modern humans, trying to trace how all humans go back to a single ancestor. You can use that same sort of family-based study to find who is the one individual that could make this third cousin and this second cousin and this first cousin on this side, and map all of those relationships together and build a pedigree and a family history to identify who your suspect is. Which, if you think about it, is scientifically kind of cool, also a little bit scary, right? From a privacy standpoint. And there is a lot of legal and constitutional issues, things that are going on in that space, that would be a different TED talk about the legality of how to do that. But there are a lot of, there is the ability to do that scientifically. It works. And then when you identify that individual, you can then go back and extract their DNA directly, compare it to the suspect, and you usually get that perfect match then. But it is kind of interesting that scientific methods that are being used sort of in a research basis, trying to identify, relatedness amongst, isolated populations in the world, or trying to identify like how all humans relate back to a single original modern human, that science can then be used in today's world to figure out who committed, what serial killer is out killing people in California, or other applications of DNA that way. So that's I think it's kind of interesting that you can bring that in, and it does bring in all kinds of other social issues, but the science is real. And a lot of times, a lot of the popular applications of genetics is usually a little bit behind the research side. Like, it doesn't come into the popular side until it's really well established on the science side. And so a lot of times when people are skeptical, like, this seems new, is this real, is this true? The answer, not always, but a lot of the time, is yes, because scientists have been working on it for decades before it became popular, and you see it in some application, and a lot of the kinks have been worked out ahead of time. If that makes the methodology and the technological hurdles. And so DNA is very, very interesting in the forensic applications. there's something, I don't know if you guys have heard something called touch DNA. So like the desk that you're leaning on right now, or the chair that you got your hands on, you're leaving your DNA print right on there just from the oils on your skin cells that are sloughing off. So you can detect DNA in a lot of places, and you are leaving your DNA all over the place, even when you don't necessarily think about it or intend to. And so that has also come a long way. That's not oftentimes shown in forensic shows. Occasionally they do show that. And sometimes I'll get a phone call, is that science fiction? Can you really get DNA from just someone touch something? And the answer is yes, like you definitely can. There's not a lot of reason to, right? No one's gonna come through the table after you leave today to like try and get your DNA from where you were leaning. But it is a real thing. And it has a lot of really practical applications, not necessarily in like just seeing where you've sat earlier. But in the Midwest, there's the Asian carp, that invasive species that's coming up the Mississippi River and trying to keep it from getting into the Great Lakes, although it possibly might already be there. It's hard to know for sure. You can actually look for the Asian carp DNA in the water. It's not exactly touch, but it's the touch equivalent for fish. And so you don't actually have to see the fish to know if it's been there. You can take water samples from various places and to see, can you detect its DNA? Because there'd be no reason to detect its DNA if it hadn't already gotten there, right? And so that's one way that you can use DNA to sort of map where invasive species are going. And it's not just the Asian carp. There's all kinds of plant and other invasive species that we have to worry about in the Midwest. But it's kind of a cool forensic related application of DNA. And so that we have enough time for everyone to extract their DNA, I'm gonna finish up by talking about DNA and COVID. We're not going to delve into all of there's so many facets of COVID that we could talk about that you could probably have a whole session on that. But we're going to just think about it from a genetic perspective, which doesn't get enough coverage, I don't think, in the popular press. But if we think about, so I'm sure everyone's heard of Delta, right? The variant that is now sweeping the nation and causing all kinds of problems. But Delta, and just to clarify, so Delta is like the name of a mutation profile that emerged. There isn't actually like a Delta mutation. Geneticists have all kinds of weird naming ways to name mutations. Sometimes, usually it's by base pair and it gets really complicated. There's like 3 different systems that you can use. So you could have 2 people talking about the same mutation, but using different names for it, which is always really confusing and fun. And so the World Health Organization tried to make that easier by giving these Greek letter variants so that people could be talking about, profiles without having to try and use the same terminology when talking about mutations. So there isn't necessarily a single Delta mutation. It's a mutational profile that refers to sort of like a family that has emerged from the original coronavirus. And so that mutation profile, from a genetic perspective, was somewhat inevitable of coming out, right? Because viruses replicate, that's what they do. And from the virus's point of view, it wants to make more virus, right? And so the more that it's replicating, it's gonna be trying different mutations to see, can it spread more quickly? The only way to sort of stop that process from happening, right, is to reduce the number of times that the coronavirus is multiplying. That's it, because it's a natural process. Viruses multiplying are always going to be mutating. Most of those mutations are going to be, neutral, no major effect. But if you can find, from the virus's perspective, it's trying to sort of almost do the equivalent of winning the lottery, right? It wants to find one where it'll let it multiply more quickly, infect people more easily, like spread more, exist outside of a human longer. That's what the virus is trying to do. And obviously we're trying to do the opposite, right? But if you think about it from a genetic standpoint, the more the virus is out there multiplying, whatever the context, whether the person is vaccinated, not vaccinated, whether the person is in Africa or India or Germany or wherever they are, every time that virus multiplies or replicates, it's going to be having mutations. Most of those mutations are probably just neutral or, by chance, and it just washes right out. But it's sort of just kind of like keeps playing the lottery. And when it hits a mutation that gives it an advantage, all of a sudden that strain is going to spread really quickly. And that's what's happening with Delta. That's, if you've heard about Lambda and Mu, those are other profiles that have that potential. From a genetic standpoint, now that Delta is spreading so rapidly, there's the chance that there could be another mutation that happens on the Delta profile that, if it makes it less responsive to our treatments that we have, or easier to go around the vaccine for those of us that are vaccinated, that could be a really big deal, right? From the virus's point of view, it would be like the lottery winner. From our perspective, sort of the worst case scenario. But the difference is, the more it plays, the more it's replicating, the more chance it is for that to happen, right? You can't win the lottery if you don't play, right? And I'm sure people in here know you could play the lottery a whole lot and you could never win, right? That's also very true. But the more chances you have, the more opportunity from a genetic perspective that mutation that they could hit on, that the virus could just hit on a mutation that would be really problematic for us. And that's one of the things when we think about mutation has a really negative connotation, right? No one wants to be a mutant, right? But the idea of mutations happening is a supernatural process. We all have mutations, right? When we're out in the sun, it causes mutations occurring in our skin cells. Like every time our DNA replicates, there's always mutations that occur. It's just the vast majority of them have no consequence. But when you have a virus that is multiplying so rapidly and moving through the population, that's where that risk comes in, which is why geneticists, epidemiologists are sort of trying to say the less people that have COVID, the better, right? Because the more, and it doesn't matter, we're not just talking about the United States. If COVID is running rampant in Brazil or Egypt or Turkey, because of how interconnected our world is, it's in some ways doesn't matter where that sort of lottery type event happens, it's going to eventually affect all of us. So hopefully when you hear about the coronavirus and the Delta mutant and other mutations that might be coming down the line, you're gonna have a little bit more familiarity when you think about it, because it's really just almost like a game of chance, like Lotto, that a mutation is going to come that's going to cause us problems. We'll just hope that by keeping the levels lower, that will reduce the odds that that type of mutation can occur. All right, so I've been talking for a really long time, and that's probably you guys are tired of hearing me talk. So for those of you that want to participate, you're going to be able to extract your DNA. And this is something that will not use any expensive or exotic scientific chemicals. We're going to be using Gatorade, dish soap, and rubbing alcohol. And that is actually all you need to be able to extract DNA from any living thing. I actually did this once with some kids, and the next day when they came back, they said, I did that with my cat. I extracted their DNA. And you'll see the first step where you have to swish the Gatorade around and spit it out. I was like, how did you get the DNA from the cat? And she said, oh, I just shook the cat and put water in the mouth and shook the cat. And I was like, oh, okay, something tells me the cat did not like that very much. But it does work. So you guys are going to use cheek and saliva cells. I'm going to stay away. And if you're uncomfortable taking your mask down and swishing and spitting back into a cup, that's perfectly understandable. But the reason why we use Gatorade is because, and it doesn't have to be Gatorade. It could be any type of sugar water or salt water. When I do this with college students, I make them use salt water. I'm not nice to buy them Gatorade. But what you don't want to use is pure water, because you want some ion in the water to help extract your cells intact. If you use pure water, a lot of the cells in your mouth will actually burst open while they're in your mouth. And while we do want to burst your cells open to get to the DNA, we want to do that in the test tube. So by putting either sugar or salt in the water, it helps actually extract the cells out intact. It also serves as like a little mini exfoliator if you've ever had like those beads in your soap. So when you're swishing it around, it'll scrape some additional cells off your cheek so you get even more. So it helps you get more cells, and it also helps keep those cells intact. You do not need a lot. You should just have a little bit of like the very bottom of the cup should have a little bit of Gatorade in there. And so what you're going to do is you're going to lower your mask, put it in your mouth, swish it around. I generally, to get the best yield, you want to kind of swish it around for 1 minute, which doesn't sound like a long time, but if you've ever done something for a minute, it can be a long time. So I would say just kind of do it for as long as you're kind of comfortable, and then spit it back into the cup. And then we'll go on to the next step. So if you want to try to do it for a minute, you can kind of watch your watch. I can make an announcement however works best, but a minute can be kind of a long time. Depending on when the last time you ate or drank is, you may get sort of a mixed yield, but it shouldn't affect anything. Just you want to get as many cells as you can, and then you'll spit it back into there. All right, you should have in your cup a mixture of probably today's breakfast, anything you ate in the tailgate earlier, and some of your cells. If it's colored because you drank orange juice or something colored earlier, that's okay. It's not going to mess anything up. As long as your cells are in there, that's all that matters, and they should be. And so in the test tube, you should see there is some dish soap in there. I think everyone has orange, although I buy whatever's on sale, and so it sometimes changes the color. The color of the dish soap means nothing. And so if you want to do this at home, pick whatever dish soap you have. As long as it's the liquid soap, it's all good. And so you should see on the test tube, there is numbers on the side. And it doesn't matter exactly how much dish soap is in there. But what we want to do when you pour your cells and your spit in is you don't want to fill it all the way up, because we're going to add more things into the test tube. So I would say aim for a total of around 10 milliliters. If you don't have 10, that's okay, because maybe you swallowed a little bit of the Gatorade, that's okay. But I would try to stop it around 10. Go ahead and open your test tube, pour in your spit mixture, put the lid back on. And if it's like 11 or something, that's fine. Just don't blindly dump the whole thing in and fill the tube up. That's what you don't want to do, because you can't take it out afterwards. So once it's in, it's in. And then you'd put the lid on. So make sure the lid is on securely first, right? And so when you look at it, you should see that the soap and your spit mixture isn't very well mixed. You can see parts that are really light or parts that are really dark colored. And so that is called sort of a heterogeneous solution, right? It looks different. We want a chemically homogeneous solution, which means you want it to all look the same. So you're going to shake it up. With soap, sometimes it's easier to actually turn the tube upside down because it doesn't move, like just straight shaking doesn't always get it really well mixed. You will see, and the reason why you're shaking is because DNA is generally found in the nucleus. It's found in the mitochondria as well, but we won't talk about that today, but it's mostly in the nucleus. And so to be able to see your DNA, you need to open up the cell, which means to disrupt the cell membrane. And you need to open up the nucleus, which means disrupting the nuclear membrane. And both of those membranes are made of lipids. And lipids are kind of interesting molecules in that they have both a hydrophobic and a hydrophilic end. And what that means is there's one end of the molecule that likes water, one end of the molecule that does not like water. And that's how it holds the membrane together. Soap will cause the membrane to spin around really quickly to try to align the end that doesn't like water away from the soap, right? Which is why soap causes suds when you wash your hands generally. When you're washing your hands, if you think about it, and again, this is sort of the science perspective, when you're washing your hands, you are actually causing the bacteria on your hands to explode because their membrane is disrupted because the molecules of the soap cause it to spin around and the membrane bursts. And so you're bursting your cell's membrane in the test tube right now. And what that means is your DNA is just hanging out in the soapy mixture right now. You can't see it. If you see anything that's actually floating in there right now, that would just actually be probably your breakfast or chunks of food or something in there. But we want to make the DNA visible. And so to make the DNA visible, we're going to add rubbing alcohol to your test tube, which is going to pull the DNA out of the soapy mixture. And so once we're going to come around with the rubbing alcohol, what you're going to want to do is not shake it anymore once the rubbing alcohol goes in. So the rubbing alcohol is less dense. It's going to form a layer sort of right on top of your soap mixture. Right now, you probably have bubbles that are right there. Bubbles are going to disappear. There's going to become a clear layer right on the top. And what's going to start to come up is your DNA, okay? Depending on how much DNA you were able to extract from your spit. And so you're going to want to watch sort of the layer right where the clear layer and the soap layer meet is where you're going to want to focus your attention. People say it looks almost like a floating spider web. Some people say it looks like snowflakes. If you have a lot of DNA, you can sometimes see the string kind of connecting it together. Sometimes people say it looks like frozen bubbles. There's lots of different descriptions. So you're going to want to focus on that. If you drop your tube or you shake your tube or you lay your tube down on its side, if you didn't have a lot of DNA to start with, it's going to disappear. So you'd have to add rubbing alcohol in again, which we can do. But you're going to want to try to keep it upright till you see your DNA, because otherwise it'll disappear. So we're going to zip around. If you can, when you see us coming, just open up the top of your tube. We'll just shoot in the rubbing alcohol, and then you can put the lid right back on. All right, I think most people have the rubbing alcohol. We've got last corner still getting it. If you ate or drank right before this, probably you didn't get a ton of DNA that's precipitating right away, but you still should see something. Usually it starts off, the frozen bubbles are sort of what is the first thing that you see. You want to look right at those two interfaces where they're meeting. Sometimes if you put a dark background behind it, it might look almost like a grease cloud. If you've ever seen like a grease cloud when you're like washing dishes or something like that, that grease cloud is actually your DNA. If we sat here long enough and held it upright, eventually you would see almost like a whole jellyfish of your DNA that starts to come out. Some of you might already have that. So if we were at the lab at 23andMe, or you wanted to do a genetic test for some type of medical condition, you could actually swirl up the DNA that you see right there with like a toothpick, move it to another tube, and use that DNA for an ancestral test, for a medical genetic test. It's the same DNA that they use. You'd want to clean up the soap and the rubbing alcohol out of it, but this extraction process is the same first step that they use in medical labs, in hospital labs, in forensic labs. This is how you extract DNA. There are multiple ways to do it. Probably this isn't the most efficient way, but it does let you be able to visualize it. And what you're seeing, if you wanted to use that DNA for some ancestral test, you could absolutely do that. Just swirl it up with a toothpick and collect it. So I am sorry I didn't say at the beginning, you can always interrupt me with questions. Does anybody have any questions, whether about this earlier stuff? Okay, we've got, Yes. Not in any way, shape, or form, no. So mRNA, by definition, is super transitory. It is hard to keep mRNA stable. That's why our body doesn't use mRNA to store genes or to store information. If you go back to the Watson and Crick paper, that's why DNA was discovered, and that's the function of DNA. DNA is stable, mRNA is not stable. And so the challenge with mRNA, and the reason why mRNA vaccines are sort of new to the game, is it's hard to keep something stable long enough to even inject it into a human to make it work. That's the hard part. Of all of the things to worry about with the mRNA vaccine, it would not be that it's going to be able to stay in your body or to be able to be into your own DNA, because it's hard enough to keep it stable to inject it to begin with. So the fact that it stays stable long enough to be actionable is sort of the scientific novelty, but it is not going to just go into your DNA or disrupt it in any way. Does it go into the nucleus? I don't know that off the top of my head. Generally speaking, I don't believe so. It's transcribed out in the cytoplasm, but I don't believe it does get into the nucleus. It's working with mRNA in the lab. You have like the half-life of mRNA is so short. You're ecstatic when you can make it last like a transfection that you can see an effect within 24 hours. Like once you dissolve the mRNA, you're like on the clock trying to get it into your cells, electroporate them. Like it's very, very hard to do. So there are some things that you might want to be worried about or concerned about, but mRNA being stable or disrupting your DNA is not one of them. Yep. They can, and some viruses do. A lot of times when they go into your DNA, they're not active anymore, but they can. Yes. 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 Yes, and there are others. You can find lots of viral artifacts once you go through the genome, or things that are thought to be viruses from the past. Yes. Okay, we had a question over here.
As more DNA profiles are at database, would it ever be possible to actually identify a specific individual?
So the way those algorithms work, probably, so it would be possible to do that if that was your sole goal, right? So if you're a computational biologist or you're a statistical biologist and you're trying to do that, you could potentially do that. Find the exact lineage of that person. Like the companies that are doing that, that's not their goal. So you would never get that result from Ancestry.com or for 23andMe. But if you were to hire like a smaller firm with that exact purpose of mapping out your ancestor, it is scientifically, technically able to do that, but it's not part of sort of the general return to results. Does that make sense? Okay, any other questions about the, okay. The companies sell your DNA. That's an excellent question. So obviously the answer is complex. There's not one answer for all of the companies. Basically, 23andMe they don't sell your DNA, but they sort of sell access to it so that you can use it for drug discovery. So pharma companies can use it for drug discovery, or they use it for sort of research purposes. It's de-identified, so in theory, it's your DNA that's being used, but it's not linked to your name, address, identifier, etc. One of the things, and so genetic privacy is a really big thing. We could do one of these talks on that. That's a whole nother topic. But one of the things, and I'm a big proponent for genetic privacy, I'm very sympathetic to that. One thing that I love about 23andMe is you do not have to tell them your real name or your real address. And there is literally nothing to stop you from registering your account. And probably John Doe is too generic, right? I would recommend make up the name you've always wished you had, right? Come up with, like, you do not need to put your accurate identifiers on there. You can opt out of all of those sort of research-based studies, but you also can put a fake address on there, put a fake name, live wherever you wished you lived, and live in Waikiki. And if there's our pragmatic things that you can do to shield your identity more seriously, then they don't necessarily, they don't sell it, but it's not super private either, if that makes sense. I believe you can, and I believe you can actually ask them to remove you completely from the database.
What if they found the gene at Carp?
Yep. Right. So, this goes into a lot of other privacy laws. The short answer is no, they shouldn't be able to do that. But could they is a different story. And I think one thing I would say, just to, if anyone has that as a pressing worry, if there was such a gene, I think it would probably have been identified already, because there's been probably 20 to 25 years of really serious interrogation of the genome, looking for those single gene predictors and mapping out when there's a gene one-to-one relationship between a gene and a disease. Those things are becoming, I won't say super well understood, but mapped enough that you know what the genes are. Like the gene with, you know, BRCA1 and BRCA2, the cystic fibrosis gene, like those types of things. If there was a gene that caused people to murder people, I think would have already been detected. Probably that's going to be a lot more complex than a single gene cause. Are there any other questions? Uh-huh.
But they still had some shreds of viral RNA that was not intact, couldn't replicate. It was test positive,
Right.
and that would constitute a false positive.
Yeah, you could.
False positive is what percent of the news media are saying how many cases there are, not how many hospitalizations.
Right. So in general, false positives are going to be a little bit harder to get than false negatives, right? Because a false positive means there's some, like depending on what part of the viral genome you're detecting, you know, obviously you're not going to sequence the entire viral genome. You're picking some region to look for. And if you detect that region, it's going to ding as a positive, whether it's an intact genome or a shred or whatever the reason might be. But if you have no exposure to the viral genome, they are looking for a piece of DNA that in theory you shouldn't have if you haven't had exposure. So that's why I say false positives are a little bit, I won't say unusual, but harder than a false negative, because if it's harder, if you get a sample that just didn't get the viral sample on it, you could still be positive without detecting it. If you actually detect the virus's DNA, it doesn't tell you that it's an active virus. That's true. But it does mean you have some exposure to it, whether it's, you know, a long time ago and you've recovered, whether it's being deactivated. And so that type of distinction can really only be done by in-depth sequencing or by changing what region of the genome you're looking for. So if you're hitting a lot of false positives, you could look at a different part of the genome to maybe reduce that. That's less common. But I think in general, our testing protocols don't do a lot of sequencing. They do sort of just this PCR-based test or an antigen-based test. And if we did more broad sequencing, it would detect those types of things. It would also help us detect mutations that are occurring more quickly. And that's probably a whole nother talk, but our infrastructure on testing leaves much to be desired across the board. But any other questions on the experiment? I know it's just about time. I'm happy to stay here and answer any one-on-one questions someone might have. And I would encourage you, the experiment that we did today, you can throw it away or take it home with you. It doesn't make any difference. But everything that we used, sugar water, dish soap, rubbing alcohol, probably things you have at your house right now. You can impress your friends, neighbors, kids, grandkids, whomever you might want to impress, and show them their DNA. I do not recommend the cat because I don't think they'll participate very nicely. But it would work if you are able to get the cat's saliva. And again, we have some brochures in the back. Please feel free to spread the word about the DNA Center. We are reopened after 18 months of hiatus. Our summer programs are one of probably our most popular things. So we have summer programs from grades 7 and up. Actually, we have some elementary for day programs, but starting in grade 7, we offer residential opportunities. So people have come from all over. They get to stay in a Notre Dame dorm. They get to be in the lab here. They get to work with all kinds of cutting edge biology experiments that are normally done by college freshmen, but they get to do it at their level. It kind of makes science a lot more exciting than the memorizing vocab and textbooks that they might get in their school. We've gotten really great reviews from parents and participants. And so spread the word even for far family that maybe aren't local, but might enjoy that type of opportunity. I built the program around what I wish I could have had exposure to when I was a kid and never did. So it's a very unique program, and it's pretty hands-on and well received. So if you're interested in that, you can get on our mailing list. Registration generally opens in January, and enjoy the rest of your football weekend.